Well, it's really nice to be back and see many of you again and get excited again about all the amazing work that's going on here. And thanks for the chance to present a little bit about my work. So I'm interested in the neural basis of mammalian movement. Amazing complexity and agility of mammalian movement is really critical to the evolutionary success of many species. And I think this is really well illustrated by the acrobatic feeding behavior you see here in the harvest mouse. But success in domestic environments can also require sophisticated motor behaviors, like this one here, this cat sneak attack. Sneak attack, yeah, right there. But unfortunately, our mechanistic understanding here remains very limited. We simply don't have very good ideas about how neural activity is bouncing around the motor system in real time to generate movements like this. So the work in my lab uses the mouse, which is relatively new as a model for motor control, to answer basic questions about how the motor system gives rise to movement. So with my 5 minutes, I'm just going to tell you about the project that I'm working on, which is furthest along at this point, that we're starting to write up. Here, a graduate student, Claire Warriner, has been working with me using the mouse, examining how the motor cortex helps enable the simultaneous co-contraction of antagonist muscles. And here she's using the mouse to really build upon some ideas that have come from previous work in cats and primates. So we know that the rotation of joints requires the alternating activation of antagonist muscles in order to rotate that joint. There's, of course, like the triceps and the biceps that help rotate and extend, flex and extend the elbow. But it's the simultaneous co-contraction of antagonist muscles that's necessary to stabilize joints against rotation. And this is important during many movements. For example, when we're trying to very precisely position our joint angles, to very precisely position our limb, like in the cat here when it's planning its sneak attack. In thinking about underlying mechanism here, one idea from the literature is that the cortical output driving antagonist flexor and extensor muscles is simply combined together in order to enable co-contraction. But there's also evidence that cortical output does something unique to facilitate co-contraction. There are intrinsic spinal circuits that help ensure alternation during joint rotation, antagonist muscle alternation joint rotation, like the classical 1A inhibitory interneuron. And there's some evidence actually from human studies that imply that cortical influence mutes this inhibition to facilitate co-contraction. Apparently by activating presynaptic inhibitory interneurons that inhibit sensory afferent terminals, and by inhibiting the 1A interneurons. And using the mouse, Claire has begun to dig into and test some of these ideas. So the first thing Claire did was develop a behavioral paradigm in which mice will alternately do an alternation behavior and a behavior that requires co-contraction in order to get rewards. So in one of the task phases, the animal has to iteratively turn a small wheel with its right paw in order to get rewards. And in the other task phase, the animal holds a stationary handle, and then rewards are dispensed when it simultaneously co-contracts its biceps and triceps muscle. And in each behavioral session, there's an epoch of the co-contraction behavior that's flanked by two separate alternation epochs to allow us to control for changes across time. Here you can see average muscle activity of the muscles that Claire records from in this behavioral paradigm, but our focus here is really on the biceps and the triceps that you see here in color. And in activating forelimb motor cortex with the injection of mucimol causes a dramatic reduction in task performance for both of the phases of the task. So now if pathways activating flexors and extensor muscles are just combined or co-activated during co-contraction, then we'd expect to see similar patterns or similar correlated patterns of flexor and extensor related activity in motor cortex during alternation and co-contraction. But this is actually not what we see at all. So Claire used silicon probes to record from populations of neurons in the deep output layers of motor cortex. And then she used behavioral triggered averages of the activity of all of these neurons to fit the averaged activity of simultaneously recorded muscles. And she found that models fit to the muscles during alternation, or the first alternation epoch, generalize well to the second alternation epoch, but actually do not fit data from co-contraction well at all. And she can go the other direction and fit models with the data from co-contraction. These don't generalize to alternation well at all either. We can fit these models using either all the recorded neurons or just the subset of neurons that have relatively wide waveforms, which should be predominantly pyramidal neurons. And in both cases, we see the same sort of fit quality, quantified here in terms of the R² values. Now, the key observation here, though, is that the neural activity subspaces inhabited during alternation and co-contraction are quite different. So if descending pathways activating flexors and extensors are simply co-activated during co-contraction, we'd expect the same subspace to be inhabited during these two tasks, but that's also not what we see. So the overlap in the activity subspaces for the two alternation epochs is about 50%, so there is some change across time, but the overlap we see when we compare the activity subspaces from alternation and co-contraction is much lower. It's down around 20%. And that 20% value is actually on par to the amount of overlap we see in the subspaces inhabited during alternation and when the muscles are actually completely inactive. Now, to show the activity subspace differences we see here hold for specifically the activity which is muscle-related, we projected all the neural activity down into a subspace defined by those linear models that I told you about in the previous slide. So each of those linear models basically just defines a direction through the full neural activity space. Collectively, then, the models fit to several different muscles, define a low-dimensional subspace, and when we project all the motor cortical activity through that subspace and do the same sort of analysis, we see the same sort of trend here. And in fact, this trend also holds for activity specifically in corticospinal neurons, which are the relevant output neurons here. We made GCAMP6F fluorescence measurements from the apical trunk dendrites of corticospinal neurons to estimate their calcium fluctuations and spiking. And we see the same differences between activity subspace overlap in terms of the calcium and the spiking for these cells specifically. So collectively, these data seem to indicate that motor cortical output is engaging spinal circuits in a unique way to enable co-contraction. And we're currently analyzing similar data to test this prediction that motor cortical output specifically activates presynaptic inhibition during co-contraction. And here we're using our transsynaptic rabies approach to measure activity specifically in the subpopulation of corticospinal neurons that synapse directly onto the presynaptic inhibitory interneurons. With that, I'll just end quickly, say thanks to all the people that helped here, including Claire Warriner, who did all of the beautiful work. And thanks again for the chance to present. Thanks.